Now let's focus on how to build a genetic library. So we're going to use the concept that we learned in the previous lecture. We're going to use something called a restriction enzyme to build a genetic library. Now, what exactly is a genetic library? Well, a genetic library for some given organism is basically a collection of all the different types of genes that are found on the DNA molecule for that particular organism. So let's take a look at the following 7 steps that basically describe how to build a genetic library. And for simplification purposes, we're going to examine a hypothetical organism that has a DNA that only consists of 4 different types of genes. So let's take the following DNA of some hypothetical organism that consists of gene 1, shown in green, gene 2, shown in purple, gene 3, shown in red, and gene 4, shown in orange. So as always, we're dealing with a double helix DNA molecule that consists of these 2 individual single strands that run in anti-parallel direction. So the first step in building our genetic library is to use these restriction enzymes that we spoke of in the previous lecture. So we basically pick a special type of restriction enzyme that is capable of cutting this DNA molecule exactly at the locations where each gene begins and the other gene ends. So we cleave it right here, we cleave it right here, as well as right here. So at the end, we basically produce the following 4 individual genes. So step number 1, we cut our DNA molecule of that organism with a restriction enzyme. Now, the next step is to basically take a plasmid. So the next goal is to take some sort of vector, some sort of carrier that can actually hold on to that gene. And the vector carrier we're going to use in this lecture is a bacterial plasmid. So we take a bacterial plasmid and we place it, we mix it with these genes, and we make sure that we also mix the plasmids with the same exact restriction enzyme. And the reason we want to mix those plasmids with this same restriction enzyme is to make sure that the sticky ends that are produced are complementary to the sticky ends that are produced within this particular case. So that once we mix the bacterial plasmids with these genes, those genes can basically use their complementary sticky ends and attach onto that bacterial plasmid. And at the end, we also add DNA ligase to basically make sure that our bonds are fully formed between these genes and our plasmid. So at the end of step 2, we now have these 4 different types of plasmids where each one of these plasmids carries its own gene. So this carries the green gene, this carries the purple gene, this carries the red gene, and this carries our orange gene, where these sections here are our genes, and these brown sections are the rest of the DNA sequence of that particular plasmid. Now, also notice that the plasmid comes with this special blue section. And what the blue section is, it's a special gene that codes for a special protein that gives the bacterial cell resistance to drugs, resistance to antibiotics. And that will become important in just a moment in step 4, as we'll see in just a moment. So let's move on to step 3. Once we form these plasmids, we now want to insert these plasmids into actual bacterial cells. So what happens is we take these plasmids, we mix them with bacterial cells, and some of these bacterial cells uptake these plasmids via the process of transformation. So what happens at the end of step 3 is we have this collection of bacterial cells, and some of these cells will have the plasmids and some of them will not. Now, another thing that I should mention about these bacterial cells is these bacterial cells do not actually have resistance to antibiotics unless they have a plasmid that has the gene that codes for a protein that gives that cell resistance to drugs. So to see what we mean, let's take a look at the following diagram. So this cell, and this cell, and this cell, they all took up those plasmids, but this cell did not take up the plasmid. Now, because these cells have the plasmids, and each plasmid contains that blue gene, these 4 cells will all have resistance to antibiotics. But this last cell that did not take up the plasmid, it will not have resistance to drugs. And so if we expose this last cell to antibiotics, it will die off. In fact, that's exactly what we do in the next step, in step 4. So in the next step, what we want to basically do is we want to be able to distinguish between those bacterial cells that did take up those plasmids and the ones that did not. And so what we do is we take all these plasmids and we take them and place them onto a special petri dish that contains not only a nutritious fluid but also antibiotic medium. And so what that means is once we place all these cells onto this antibiotic containing medium, those cells that took up these plasmids will have that blue gene that gives it resistance to those drugs, and so they will not die off. But on the other hand, those cells that did not take up those plasmids will die off because they won't have that special blue gene. So this cell, and this cell, because they did not take the plasmids, they will basically die off. But this cell, and this cell, because they have these blue genes that gives them the resistance to antibiotics, they will not die off, they will survive. So now we basically have all these cells, and we know that all these cells carry the plasmid. The next step is to basically take each one of these types of cells. So let's suppose we take this cell that carries this plasmid, and we take this cell and we place it onto a petri dish that has a nutritious substance. And what happens is this cell now divides via binary fission, producing millions and millions of these bacterial cells that have identical copies of these plasmids. And we follow the same exact step with the second type of cell that contains the second plasmid. Then we follow that with the third cell that contains the third plasmid, and the fourth cell that contains this fourth plasmid. And so at the end of the step, what we have is a colony that consists of millions of these identical plasmids. And we have a colony for each and every one of these different types of cells. So once we form the colony of cells in each one of these petri dishes, so now we have 4 different petri dishes, we can basically lyse the cells, and by lysing the cells, we basically release those plasmids, and now we can expose the plasmids to these same restriction enzymes. And what that does is it breaks down the restriction enzymes and it releases each one of these genes. And now we're dealing with not a single gene, but we have millions of each of one of these genes. So we have many of these green genes, we have many of these purple genes, we have many of the red genes, and we have many of these orange genes. And so now we have a collection of these genes that constitute the DNA for this particular organism. And that's exactly what we mean by a genetic library. A genetic library is basically a collection of all the different genes that are found within that particular organism's DNA. And this is how we basically create a gene library. Now, once we create the gene library, now we have the collection of all these genes, and we can conduct many different types of experiments, as we'll see in the next several lectures.